Hello, and a very warm welcome to everyone to our today's webinar on challenges and solutions, development and validation of Omalizumab clinical assays. I am Anvesha Bhattacharya and I represent Vida Clinical Research. Vida Clinical Research Limited, together with its subsidiary Bioneeds India Private Limited and its joint venture Ingenuity Biosciences Private Limited, together referred to as Vida Group, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovator, biosimilar, and generic drug development programs for our global clientele. Together, we serve clients globally in pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal nutraceuticals, medical devices industry. As your facilitator today, I would like to introduce you all to a couple of points as we begin in this session. This session will have a presentation in the beginning, followed by a Q&A session. We have enabled our Ask a Question features. It's right on your screen, where you can drop all your questions related to this webinar right in there. So now moving forward, I would like to introduce you all to our panelists for today's session. Our first panelist is Jessie Riggs. She is a bioanalytical principal investigator at Somru Bioscience, Prince Edward Island, Canada. She serves as a scientific and technical lead for the development and collaboration of large-scale client collaborations and projects. She is responsible for the development, validation, and application of analytical methods to support biologics, biosimilar, and vaccine studies to comply with scientific regulatory requirements. Jessie has more than 9 years of experience in professional research of regulated bioanalysis to support clinical studies of biologic products. She brings a comprehensive knowledge of bioanalysis and hands-on operational experience in delivering scientific and service excellence to pharma and biopharma companies. So our next panelist is Shahid Hassan. He is a global project manager at Somru Bioscience. He is responsible for team representation and project management responsibilities supporting cross-functional R&D and CRO projects at Somru. He holds a master's degree in computer science and has more than 5 years of automation and project management experience. So without any further delay, I would like to call upon Jessie to begin the session.
Hello, good evening. Just going to share my screen here and we can get started. So today we're going to be discussing the challenges and solutions series for the development and validation of Omalizumab clinical assays. We'll discuss the Omalizumab overview, the structure, mode of action, and characterization, along with the challenges and solutions faced in the assessment of the Omalizumab clinical assays for the Pharmacokinetic, the total IgE, the free IgE, Immunogenicity, and Neutralizing antibody assay. Omalizumab is a recombinant humanized monoclonal antibody against human IgE. It is derived from a murine monoclonal antibody, which was humanized to produce omalizumab in its present form. It contains 5% non-human amino acid residues, which make up the complementary determining regions of the protein. These regions would be in this white area. Structurally, omalizumab is a glycoprotein, and it is produced by recombinant DNA technology in mammalian Chinese hamster ovary cell expression systems. The mode of action of omalizumab. Omalizumab mode of action inhibits the binding of the IgE to the high affinity IgE receptor on the surface of mast cells and basophils. Reduction in the surface of bound IgE on the receptor-bearing cells limits the degree of the release of mediators in the allergic response. Treatment with omalizumab also reduces the number of the receptors on basophils in atopic patients. So getting into our assays, we're going to get started with our pharmacokinetic assay development, our omalizumab PK assay. So here you can see our omalizumab PK antibody sandwich assay. We are capturing the omalizumab with the drug target IgE. We are introducing our drug in calibrators, QCs, and samples in serum, and detecting with a specific anti-omalizumab conjugated with HRP. Our sample matrix is serum, and the expected Cmax for a 300 mg dose is approximately 33 microgram per mil, which is around what we were optimizing for. And then for a larger 600 mg dose, approximately 67 micrograms per mil. So in our initial early development, once we decided on this format, we were running our assays and we observed some significant drift in signal from the front end of the plate and to the back end of the plate. So the back end FAR QCs were consistently having lower signal. To confirm this, we ran a couple assays with 8 replicates of each QC level across the plate from left to right. And you can see here in this graph that it was significantly dropping as we went from left to right. So on the right hand front side of the plate, signal was where we expected it to be, and by the time you were on the far end left side of right side of the plate, the signal was significantly dropped, especially in the high QCs. So to address this, we were looking into our various incubation times. So we wanted to make sure that every reagent was able to bind at its maximum amount. And so we optimized using various incubation periods for each step of the assay. So our coding, our blocking, our sample incubation, and our detection. So we did see improvement of this by combining larger incubations for most steps. So we ended up with an overnight coding, a 2-hour blocking, and a 2-hour sample incubation. We did not find that increasing the detection improved this. However, we were able to confirm when we ran 10 replicates across the plate from far left to far right, and at each of the QC levels, and we could see that this drift had been corrected. So once we were able to correct this drift, we moved into running this, getting it ready for some accuracy and precision. When we started to do this with multiple analysts, we were seeing a high level of variability from analyst to analyst, and also run to run by one analyst. So when we initially looked at this, we did find that the analysts were utilizing our best methods for reagent storage, reagent aliquoting, everything like that. And they were also all using a standardized pipetting technique. So there was no variability in how the assay or reagents were being treated from assay to assay or analyst to analyst. So we did determine that it was inherent to the actual assay, this variability. So we were looking for solutions to correct this. We did realize that the variability was highest on the high end of the curve. So our top calibrators, and our curve range had been wider than it was required to be. So we did decide to truncate our curve to the required range for our calibration curve. So we adjusted that from 2500 to 1200, keeping the bottom sensitivity of 40 nanograms with an anchor of 20 nanograms the same. Once we did this, we did see that the variability was strongly reduced. However, our signal was a little lower. So we did also increase our HRP and repeated our precision with 3 analysts over multiple days. And we were able to determine that the variability was within an acceptable range. And so we were able to move forward into our accuracy and precision of this assay. So once we completed our accuracy and precision, we did find that we had our functional assay with acceptance criteria being met. And here you can see our accepted accuracy calibration curve with our standard deviation error bars and our mean total mean bias CV and results for our QCs. So at this point, we turned this into validation team, and we were able to validate and are ready for sample analysis for the pharmacokinetic Omalizumab assay. Once we were finished with our Omalizumab PK assay, we got turned towards our PD assays, being total IgE and free IgE, to support the pharmacokinetic assay. So with our total IgE, we are also using a ELISA sandwich format. We are capturing with a polyclonal anti-IgE, which is not specific to the IgE receptor in which Omalizumab binds to. So it's going to be binding with other parts of the IgE to be sure that Omalizumab is still able to bind to IgE and have the IgE be captured in the assay. Once the IgE and or Omalizumab it was captured, we are detecting with a monoclonal anti-IgE HRP, also not specific to the IgE receptor. And this assay range is between 2 and 2,000 IU per mil. So one of the first challenges we saw with the total IgE is that our calibration curve is in buffer due to endogenous IgE. So the accuracy of our QCs coming in was being affected with serum interference. So due to the endogenous nature of the IgE, we needed to run our calibration curve in buffer. And so we had to be able to, but however, our samples are going to be in serum. So it is unavoidable that our QCs need to be in serum. So what we were able to do was we turned to a Samru proprietary blocking assay buffer. We have used this previously and developed this previously to deal with serum interference. And so once we were able to do this and use this blocking buffer, we were able to improve our accuracy of our serum QCs. And you can see that the accuracy was vastly improved once we changed our blocking buffer. And we were able to recover our IgE in the serum. So our IgE calibrator is qualified as in IU per mil. To do this, as IgE is reported in IU per mil, we had to convert our calibrator from a nanogram per mil to an IU per mil. And to do this, we used an international standard from NIBSC and ran parallelisms with our IgE in various dilutions in the standard curve for the NIBSC IgE standard in the linear portion of that curve. We ran this a minimum of 3 times with multiple analysts, and were able to interpolate and get the mean of the IU per mil stock of our IgE, calibrator IgE, and convert that into IU per mil, and then continued to develop our assay in IU per mil. So again, with our accuracy and with dealing with endogenous IgE, so due to its nature, it is going to be invariably constant in any serum. And for our QC preparation in serum, we have to take into mind the IgE that would already be present prior to our contrived samples. So we decided to move forward with an additive method where our serum IgE level is determined over multiple assays by multiple analysts and a mean taken. And so our serum is then given its endogenous IgE value. And then to prepare our QCs, we kind of spike on top of that to get the desired QC concentration. So my example is we determined our QC serum endogenous level to be 75 IU per mil. But if we wanted our QC IgE to be 175, we actually would add another 100 IU per mil of IgE to obtain that. So once we do obtain our desired serum QCs, that QC level is how the accuracy is determined by bias from that as compared to the calculated results. So with total IgE, one of the biggest concerns is that we are measuring all of the IgE. So the total amount of IgE, so whether it's bound or not bound to omalizumab in the system, we need to make sure that we're measuring all of it. So to confirm that, we actually introduce excess omalizumab, and during this assay design, we introduced up to 50 microgram of omalizumab as it was above our expected Cmax. And we were able to confirm that their IgE was measured accurately. So our biases were correct, and there was less than 20% variability between samples without QCs without IgE and with IgE and no omalizumab, and without IgE, or with IgE and with omalizumab. Sorry. So drug. IgE without drug and IgE with drug, we had less than 20% difference, and our accuracy and precision was also well under 20%. So we were able to confirm this in multiple runs as well at all of our QC levels that we were measuring all of the IgE. So at this point, we took our IgE assay and it turned it over to our validation team. So for our next PD assay, our free IgE is quite the opposite of total. For free IgE, we need to make sure we are measuring only free IgE. If it is bound with omalizumab, then we should not be able to measure that IgE. So for this is also a ELISA sandwich format, where we capture and coat with the specific IgE receptor that omalizumab binds to, and sorry, the IgE binds to from omalizumab. And so we would introduce our IgE, and then we were detecting with an anti-IgE HRP. And so in this, when omalizumab is bound, it is unable to be captured by the receptor, and therefore only free IgE should be measured. So some of our initial steps were to optimize our incubation time, our buffers, bound and unbound IgE, and our development included accuracy testing with the theoretical IgE. So our next slide will get into our theoretical IgE. So for theoretical IgE, we needed to be able to measure IgE accurately and confirm that we are not measuring anything bound. And so we used a formula here that would tell us what the expected amount of IgE unbound to the drug would be in the specific amount of drug. So our formula here is looking for free IgE LF. And we would need our total IgE amount introduced into the system. We would need the total amount of drug introduced into the system, as well as the equilibrium dissociation constant of IgE and omalizumab. So with those numbers, we are able to calculate for the expected theoretical free IgE. And then with that calculation is how we were determining our accuracy when drug was in the system for the recovered calculated amount. So also, when running QCs in these assays, we do want to make sure that they are always being measured. So in addition to our regular QCs, we are also introducing a different type of QC, a free QC. So these would be the HQC2 and the MQC2, which have a drug. So that would be showing in comparison to the standard regular QC, the addition of drug is going to bring that concentration down into the expected range, and that accuracy also calculated from there. So with this very specific theoretical calculation and this dissociation constant, there was a lot of very specific optimizations that needed to happen. So we were initially testing our QCs in buffer to make our calculations to ensure that way we wouldn't have to take into account all of the endogenous, and we could simplify our initial optimization. So with this being the type of assay with this coded receptor, one of the things we were testing was the amount of sample time on the plate. We found that sample on the plate was optimal around 30 minutes. And we also have to take into account our sample dilution. So in the free IgE, the calibration curve is also still in buffer. And of course, our QCs need to be in serum. And with our QC for the free IgE did not account for the MRD. So we were having to account for our MRD minimum sample dilution into our calculation as well. So the next step was to also optimize our sample incubation time, meaning the sample and the drug, to ensure that we reached equilibrium. So meaning that dissociation constant has been actually achieved. And once we reach equilibrium, a longer incubation shouldn't matter, but we did have to determine what the minimum sample time was to make sure that we did reach that equilibrium dissociation constant for our calculations to work. And once we were able to do all of that, we were performing accuracy and precision of the free analyte measurement. So this data is from our determining of that dissociation constant equilibrium. So we tested many different sample and drug incubation times. And here from 30 minutes to overnight, we did determine that 2-hour incubation was required to reach that minimum equilibrium time point. And we were with our theoretical calculation, and at this point we were in serum, so we were having to include and measure the endogenous amount as well. So we were including an NC for that calculation, and you can see that our 2-hour incubation was the minimum, and after 2 hours we continued on into overnight, and that was not affected as it is an equilibrium constant. So to also ensure that it is the free, we had to introduce drugs at many different levels. And to confirm that everything is calculating correctly. So we introduced drug, omalizumab, at 0, 10, 25, and 50 micrograms per mil, again, going over our expected Cmax. And here you can see that with the introduction of drug, our signal is the more drug being added, the lower our signal is. And we were able to confirm that only free IgE was being measured, and it was accurate beyond 50 micrograms per mil. So yes, at this point, we considered developed, and we were able to pass off into our validation team. Okay, so our omalizumab ADA assay format, we have developed a high drug tolerant speed method. So speed meaning solid phase extraction antibody. And so we were able to develop this specifically for omalizumab, and we were able to utilize this due to the high level of omalizumab in trough, expected high levels of omalizumab in trough. So here, the format is to add your sample, and there is an initial dissociation, and your biotin drug is added. So at this point, the biotin drug is added in excess, meaning it will outcompete for your ADA from any drug in the system. And then from there, that's incubated overnight. The next day, this biotin drug and sample mixture is added to a streptavidin plate. And so that will pull the biotin drug and the ADA out of the system onto the streptavidin plate and incubating there. So then at that point, the sample is dissociated again with acid, and however, the biotin strep binding is much stronger than just the protein binding between between the drug and omalizumab. So omalizumab is able to be released from the biotin drug complex, and so that is then transferred into another plate. So that plate is actually where the acid neutralization happens, and our ADA is then detected there. So this has a double acid dissociation step, and it's highly optimized in SAMRU. So one thing that we needed to consider in this speed format versus other speed formats that we've had to develop is that IgE, again, is going to be present in all of our sera. So we needed to ensure that this IgE in the sera wasn't going to impact our biotin drug in any of the complexing that was required. And so we tested the recovery in many different IgE level serums. So we used a very high level of IgE serum, mid and low, and we were able to determine that endogenous IgE did not impact the assay. Once we had our assay functioning, we were able to determine our screening cut point in running 6 assays by 2 analysts. And then we utilized our agarist immunogenicity module for our calculations for cut point, removing analytical biological outliers and determining our cut point to be an additive cut point of 0.021. So before we get into validation, we still need to run some cut point assays so that our other development assays make sense, because that cut point is so inherent to every analysis in the ADA. So we also completed our confirmatory, and we did spike with 20 micrograms of omalizumab over 6 assays completed by 2 analysts, and again, calculated our cut point in the agarist immunogenicity module. And we confirmed our confirmatory cut point to be 81.9. So the reason for these cut points is we needed to be able to determine our drug tolerance and some additional assays in which we need a cut point to determine these. So for our drug tolerance, we have actually spiked our HQC, our HPC, and our LPC with various amounts of drug from 150 micrograms down to 10 micrograms and no micrograms. And using the plate QC, we were able to determine that the sample was still detectable and LPC at 40 micrograms per mil, which would be acceptable in our case where our samples are expected to be the 300 mg dose of approximately 33 micrograms per mil. So we were able to determine that our drug tolerance was sufficient for that. So for some ADA highlights, our positive control was a rabbit positive control with the SOMRU hyperimmunization protocol, and with a sensitivity of a minimum of 100 nanograms. We didn't really test beyond that. We did have a really good signal, so we would be more sensitive if required, but for our current needs, 100 nanograms was sufficient. There was a lot of variability previous to the speed method in our initial testing. However, our speed reduced that variability with the double dissociation, and therefore no matrix effect was observed. And our IgE was not causing any issues. So between the buffer and the speed, there was no interference. And our initially testing some drug tolerance in other formats, there was very poor drug tolerance, less than 1 microgram per mil. And we were able to determine with the ADA speed method that we were at 40 micrograms per mil drug tolerance. So moving on to our neutralizing antibody assay. So this assay is a competitive ligand binding assay, where our NAb positive samples are actually going to cause a reduction in signal. So we are testing a deoxygenated drug and adding that to a IgE coated plate and detecting the conjugated drug specifically. And so once, if there is any neutralizing antibodies present, that would cause the Omalizumab drug to not be able to bind with the IgE coating, and therefore there would be no signal. So positive samples have no signal and negative signals have high signal. And that competitive nature is proving that it is only neutralizing antibodies that will cause a positive signal. So during our development, the competitive ligand bindings are used to characterize the NAbs have been shown to provide higher sensitivity. And here is our percent inhibition for our 3QC levels, 1,500 and 100 nanograms. With percent inhibition well above 20% and our 100 nanogram. Our PC also hyperimmunized rabbit anti-Omalizumab. Yes, so for overall, for Omalizumab clinical assays, we are going to require a sensitive and robust PK, PD, ADA assays with a high NAb sensitivity. Omalizumab clinical assays require experience and expertise in working with the innovator molecule to meet these regulatory expectations. We need to design assays that are robust enough to support the entire biosimilar life cycle, all clinical phases. characterization of reagents is highly critical. Matrix endogenous effects is highly critical in our omalizumab assays. Stability, ensuring supply of reagents to reduce our batch-to-batch variability, and ensuring that our critical reagents are reliable, well-managed, and qualified. So that is it for me. Thank you.
Thank you, Jessie, for nicely covering the topics in the presentation. Now, moving forward, we will be proceeding ahead with our Q&A session, and we are going to answer some of the really interesting questions posed by our audience. So my first question to you is, in the omalizumab PK assay, did you consider the effect of endogenous IgE?
Yes, sorry. So for the PK, I didn't really get involved in the discussion of the endogenous IgE, but there was a lot of testing, as I've shown with other assays, high levels, mid levels, low levels of IgE, ensuring that that was not impacting our signal.
All right. My next question to you is, in total and free IgE, you mentioned buffered calibration curves. Is that regulatory compliant? How are the samples handled in serum?
Right. So our FDA regulatory guidance does allow for our calibration curve to be in buffer, as long as we are ensuring that our quality control samples are going to be the same as our actual clinical samples. So in our case, our samples are in serum as our samples will be, and making sure that we have many different types of QCs. So in one slide for my free IgE, we actually had 7 QCs to make sure that we are covering the basis and making sure that our samples are going to be utilized in the same way that our QCs are, considering the small extra risk of having our calibration curve in a different matrix. And for a total, very similarly, where our LQC would be in is an unspiked QC. So we're actually just using a qualified endogenous level for a QC to make sure that we are mimicking our samples in serum and matrix.
Okay. My third question to you is, was solid phase extraction necessary for the ADA?
Yes, so we did find that the solid phase extraction was required. We were previously developing another type of ADA assay, just a regular ELISA based, and it was found that IgE was interfering, and we did not have very good drug tolerance at all. And omalizumab has quite a high trough level. So we needed to make sure that we could still measure our ADA in the presence of these trough levels and higher, and any kind of level. And so we did find that the solid phase double dissociation was required, yes.
All right. I think my last question to you is, was cell-based NAB considered for this?
Yes. So initially, we were considering the cell-based NAB. However, we have found that the competitive ligand bindings assays used to characterize NAB have actually been shown to provide higher sensitivity, a wider dynamic range, increased precision, and better matrix tolerance than cell-based assays. And because omalizumab is not actually impacting or directly interacting with the mast cells or basophils, it is directly interacting with the IgE. This ligand binding at NAB assay is actually more suited for the omalizumab NAB.
All right. So thank you, Jesse, for active participation and nicely answering most of our audience questions. I would like to take a pause over here. We understand this session has kept us well engaged throughout, but due to time constraints, we would not be able to take any further questions at this point of time. But please be rest assured that we will be sending the answers to every questions of yours via emails. Also, we will be sharing across the recorded link of webinar on all registered email IDs. Lastly, on behalf of VIDA Clinical Research, I would like to sincerely thank you all for being here today and taking out your time to be a part of this webinar. We at VIDA Clinical Research earnestly look forward to host you all again. Till then, take care and stay safe, everyone. Thank you. Thank you so much, Jesse.
Thank you.
Thank you, everyone.